To announce our next speaker, which will be Oleg REVA. Professor REVA is a full professor in bioinformatics at the Center for Bioinformatics and Computational Biology at the University of Pretoria. With the MSc from Kyiv State University and PhD from the Academy of Science of Ukraine, he has held resource positions in Ukrainian, German, and South African institutes, where he advanced to full professor in 2020. His research spans functional genomics, metagenomics, epigenetics, bioinformatics, tool development, and drug design, contributing to various international research projects. His mentorship has fostered a generation of bioinformatics experts across academia and industry, with over 100 publications and significant collaborations globally. He's a founder and treasurer of the South African Society for Bioinformatics with an honorary rating of C1 and Age Index score of 29. Please, Oleg, if you can please make it full screen. Oleg, can you hear us? Yeah, it's full screen, thanks.
Okay, can you hear me now? Yes? Okay, great. Sorry, I was mute for a while. Okay, so I want to thank you very much, first of all, for letting me to participate in this wonderful meeting and to listen to a very interesting talk before and to be able to present my projects and discoveries on this meeting. First of all, I want to introduce for you what is a Center for Bioinformatics and Computational Biology at the University of Pretoria. This rather small facility where we have 3 academic members and around 25 to 30 students, postgraduate students in every year. We're getting more and more popular. More students are applying for us. We can accept only 20% of applications. And students come for us from very different departments, from biological departments, from mathematics, from computer science. So we create such a small Silicon Valley at University of Pretoria, a very creative environment for multiple projects. And our projects involve all topics from bacterial genomics, plant genomics, metagenomics, ocean soil metagenomics, human genomics, sequencing of plants, and many other techniques, design of new computer programs. So I want to apologize in advance that if you find my talk less focused than the previous talks, but this is the nature of bioinformatics. We are multi-focused. And what is common now for many studies in biology is use of next generation sequencing facilities. And just a second, I'll take a point. Okay, line, shape, it's not easy. Okay, I'll try without pointer. Okay, so what is important now is that we have big revolution in sequencing technologies. Several decades ago, we had only Sanger technology. It's a very nice technique, but that served for us up to now. But then new techniques appear, generally named as next generation sequencing technology, and characterized with massive parallel sequencing. We were able to generate a lot of sequences at the same time. And this technology start to evolve in different techniques. Some became out of date already. For a very long time, Illumina technology was served for us, was a gold standard of sequencing. And many people use it till now. But now we have third generation sequencing technology, which include ion torrent, PacBio, and technologies. And for example, in South Africa, the major sequencer providers already discontinued using Illumina because it's considered as out of date. And we use long read sequencing technologies. And PacBio is already a very general term. It's Pacific Bioscience, single molecule read technology. And now we have multiple variants of this technology. REVA replaced SQL, and we have Vega technology. It's a small apparatus, desktop sequencer. We have ONSO technology for short read sequencing, but using PacBio sequencer, which replaced Illumina because of higher productivity. And it's not just different names. Amount of data generated by different third generation sequencing technology is huge, much bigger than generated by previous versions, which include information not only about sequence, but about possible modifications in the sequences, more like epigenetic modifications. So we have another level of analysis of genomes now using these technologies. More to this, different technologies change formats of data, amount of data, and it's became rather difficult to follow up, to catch up with new technologies. This is why the use of bioinformaticians is important. Okay, let's stop. Going to next page. Okay, I can go this way. Good. So what we can use a new generation of new technologies, what for? We can use it for creation of whole genome sequences, which represent for us core and accessory genes, which can be identified in different chromosomes. This is potential. What these organisms can do. Whole genome sequences allow for the estimation of genetic and metabolic potential of microorganisms, genotyping, and inferring phylogenetic relationship between them. However, we have to remember that whole genome sequencing does not reveal how the genetic potential is utilized by microorganisms or generally by organisms. There is example of a pipeline which can be used for different to create whole genome sequences. We can use long read sequencer like PacBio. We can use short read sequencer like Illumina. We have to remember that we need different software tools to operate with these different types of sequences. This is what sometimes students and researchers ignore. That we have to check quality to perform quality control of generated reads, and we have to use different programs for short read and for long reads because of different technology. We can follow up with reference-based assembly or de novo assembly of these reads into whole genome sequences. Again, we have to use different approaches for short read, for long reads, and there is many different programs which provide some opportunities and have some limitations. We can join together the results of reference-based assembly and de novo-based assembly by using other programs like RAGTAG, and then we have to check the quality of the resulting assembly. We perform quality control. We can use CheckM2 program. And these programs, we have versions which change every year, and we have to combine them into pipelines. This is what basically we provide other departments with from our Center of Bioinformatics. We can design pipelines from multiple programs. We can create environments, computer environments to the most official run of these pipelines. And very often we can find out that there is no programs which fits very well with requirements of specific projects. And we have to write our own programs. How then we can use whole genome sequences? We can compare locations of some specific genes in these whole genomes, like genomic islands in different organisms. We can build phylogenetic trees. In this case, it's terms phylogenomic trees. Where we can compare whole genome sequences. Also, we can analyze pangenomes and some genes, identify genes, including pathogenicity, virulent genes, or antibiotic-resistant genes, which are present only in several isolates, but not present in other isolates. And we can identify locations of mutations, which can turn bacteria to antibiotic resistance. For example, this example of program, which we published before, it's our own program. We try to identify, to predict antibiotic resistance in Mycobacterium tuberculosis. And here we can use such network system to identify both to which lineage, specific lineage, this isolate belong to, and is it whether this isolate has specific antibiotic resistance. Program can accept sequence data in many multiple formats, starting from FASTQ files, it's short read or long read files generated by sequencer, or results of assemblies of the sequencer, like whole genomes or partial contigs, and analyze and return for us such dendrograms, where you can see the likelihood that given organism will be susceptible or resistant to specific antibiotics. For example, on the left, you can see a strain which was identified as belonging to Beijing lineage, and most likely it's resistant to kanamycin. But very important that our program predicts not only plus minus, resistant or not resistance, but likelihoods. How likely is that this organism can develop resistance to other antibiotics. Like here, for example, this we can make time series. We can identify from the same patient pathogens at the different stages of treatment with antibiotics. We can see that generally this organism, this Mycobacterium tuberculosis strain is susceptible to antibiotics, but it started to develop antibiotic resistance, and very soon can develop antibiotic resistance to multiple antibiotics due to accumulation of specific mutations. So program analyzed not only several specific antibiotic resistant mutations, but look for prerequisite mutations, which makes this bacteria easy to develop antibiotic resistance, and compensatory mutations, which reduce fitness cost of antibiotic resistant mutations. So in different stages of treatment, we can check what mutations are accumulated in these microorganisms and how likely that in future this bacteria became antibiotic resistant. Another application of long read sequences is metagenomics. This is a tool to study pathogens in nature. Additionally, metagenomic allows monitoring pathways of circulation of virulence-associated genetic vectors, like plasmids and transposon, or even individual virulence or antibiotic-resistant genes. Here, one example of analysis of metagenomes in collaboration with our colleagues from Kenya. We have big project now on surveillance of antibiotic resistance and pathogens in basin of Lake Victoria. And we analyzed different samples. Very top sample is from tap water. The second sample in the row is from Lake Victoria water. And green boxes, it's antibiotic resistant genes identified in these samples, in metagenomes. It's interesting that in tap water, we found much more antibiotic resistant genes even than in water taken from Lake of Victoria. It's an example that not sufficient treatment is more dangerous than no treatment at all. The same like with antibiotic treatment. It's better not to use antibiotic rather than to take few piles and stop.